in your notes, make another column here where you say, from the perspective of one glucose, and you would actually, because we produce 2 pyruvates, all those numbers are gonna get doubled, right? So we're gonna ultimately, from one, as you can see here, from one glucose, we create 2 pyruvate, which means we're gonna make 2 acetyl-CoA. We're doing this process twice. This is happening, the reason we need to get it to acetyl-CoA, acetyl means 2 carbons, and so it's gonna be a 2-carbon group that enters into the TCA cycle. The coenzyme A, I'll show you a picture of the structure. It looks like a panhandle. It's like a panhandle so you don't touch the hot part of the pan. That's kind of what coenzyme A is. It's a nice handle to hand off the 2 carbons. So that's what we're prepping to get everything ready to go into the TCA cycle. All this is happening in the matrix. So between glycolysis and pyruvate oxidation, we have to shuttle pyruvate inside 2 membranes go through the outer membrane, go through the inner membrane, all the way to the matrix. Okay, then once we're on the inside there, we have 1 pyruvate, 1 coenzyme A, and 1 NAD+. And that, again, we're gonna take some electrons, that's gonna create some of the electron carriers that will go onto the electron transport chain. The production, the output, because we're going from 3 carbon pyruvate to 2 carbon, we're gonna release out CO2. So that's how carbon is released. It's completely oxidized to produce CO2. For each reaction, we get 1 CO2. So now, total, how many carbons are left from our original 6 glucose, 6 carbons and glucose? How many are left at this point, at the very end? 4, we got rid of 2. So we've gotten rid of 2 CO2s in this process. We now create acetyl-CoA, which is here. And the other thing that is produced is NADH. This is an oxidation, so the electrons are passed somewhere. They get passed to NAD plus to produce the NADH. Okay? So let's look at the chemistry of this, of what is happening. Pyruvate looks like this. The red carbon there is showing which carbon is decarboxylated. So for a decarboxylation reaction, we're taking this and creating CO2. So now we're left with this. That gets added to coenzyme A, which has a sulfhydryl group. That's going to attack the carbonyl center and form this acetyl-CoA. And the CoA right here looks like it's small. But let me show you what coenzyme A actually looks like. This is the structure of coenzyme A. Here's the sulfur. That was the sulfhydryl group that attacked the carbonyl group. What does this look like, though? If you look at the structure of this part of it, what does that remind you of? Kind of, what does it look like? It's got, remember CON? Looks like peptide bonds, right? But then this part is a nucleotide. So coenzyme A actually is a nucleic acid. It has ribose adenine phosphate, which is attached to this chain here. And this is the structure. It's this whole big thing is coenzyme A. This is the little tiny carbon groups, the 2 carbons. So it's really like a panhandle. This is considered another activated carrier, coenzyme A, that will hold on to this acetyl group until it passes it into the citric acid cycle. Okay, so coming back to this one, it involves decarboxylation, where we get rid of that carbon, producing CO2. It also produces NADH as the electron carrier. And the enzyme that does this is called pyruvate dehydrogenase. Okay, that's going to be the enzyme. Okay. So the other thing, so that I have here positive and negative feedback mechanisms. The negative regulation, so when would we want to kind of shut down glycolysis, shut down cell respiration? Would it be when ATP is high or low? High. If we already have a lot of ATP, we don't need to continue through this. So when ATP levels are high, this enzyme gets phosphorylated, and and that actually inhibits it. So high ATP levels will phosphorylate PDH, which is pyruvate dehydrogenase, and that will change its shape so that it's no longer active. Okay? For positive regulation, when there's low levels of ATP, so in other words, when AMP is high, that's when PBH will be dephosphorylated, and that's when it is active. So the phosphorylation of the enzyme itself is what regulates its activity. Okay, any questions on that step? So now that we've talked about pyruvate oxidation, you should be able to go through the second row, and I want you to actually, for the chart, to think about from 1 molecule of glucose. So you're actually going to put numbers for 2 pyruvates going through pyruvate oxidation. Did we create any ATP in that process? Did you see ATP? No. So ATP should be 0, 0. And for CO2, we're going to put 2, for both reactions. How about NADH? 2. And any FeDH2? No. So that one is 0. Okay. So then our third step here, I think we can talk about this one and then we'll take a break after we talk about TCA. All right, so far we have acetyl-CoA, and we were preparing acetyl-CoA so that the carbons could enter into the citric acid cycle. So now each of these acetyl-CoAs is going to enter into the citric acid cycle. As it's a cycle, it's going to look like this, right? We're going to regenerate the starting compounds by the end of the cycle. So the overall goal for this is to finish oxidizing all of the carbons. How many carbons are left at this point in time from the original 6? 4. So we have to get rid of 4 carbons. We can also make a tiny amount of ATP through this process and also generate electron carriers. So as we're oxidizing, we're going to save the electrons and put them with the electron carriers. Okay, so this takes place in the matrix. For these numbers, this is for 1 turn of the cycle. So if we're talking about from 1 molecule of glucose, again, we're going to double everything. So that's why I wrote the numbers here. I guess I never erased the annotations from before. So for 1 turn of the cycle, 1 molecule of acetyl-CoA goes in with, it's got 2 carbons. That's going to combine with something that's called oxaloacetate, OAA. That's the starting compound that combines with 2 carbons from acetyl-CoA to create citrate, which is the 6th carbon compound that this whole thing is named after. So 4 carbon oxaloacetate is needed, combining with acetyl-CoA. The output, we're going to get those 2 carbons off of acetyl-CoA. So we have 2 CO2s produced. We get, for each turn of the cycle, 4 different electron carriers. 3 of them are NADHs and 1 is FADH2. So there's a total of 4 oxidation, redox reactions that are going to happen. And also we can create 1 molecule of either GTP or ATP. That's kind of cell dependent or organism dependent. But even if we make GTP, that can be converted into ATP. So that's why I put them together. Okay, so for glucose, we're turning the cycle twice. So that's why we have doubled all of the numbers. In that case, we will have a total of 4 CO2, which is all the rest of them, right? There's no carbon left after the TCA cycle. And then we have 6 NADH, 2 FADH2, and 2 of the ATP that are produced. Now, what type of ATP production did we say we're using in the citric acid cycle? Still substrate level. Yeah, we haven't yet gotten to oxidative phosphorylation. Okay, so this is what the cycle looks like. Again, lucky for you, you don't have to know all of these intermediates. This, you can kind of see what's happening in the beginning. Here's oxaloacetate, 4 carbons combined with acetyl-CoA. That gets rid of the CoA, which is going to recycle it. And that produces citrate. So we have a 6th carbon This, I will not ask you to know all the reaction types, but you probably could figure them out, right, for the most part. Citrate is reorganized. So what is it called when you reorganize something? Isomerization to get isocitrate. Then we have a decarboxylation where we lose one of the CO2 groups. And in that process, we hand off the electrons to NAD+. So here's redox. Here's a second redox reaction. So here's two of the NADHs that we're producing, and there's your two carbons. This is the step where you're producing ATP. So this is where we get, it's a phosphorylation step. We're getting a little bit of ATP. And then here again is a redox. We produce FADH2. One more redox over here. And the goal for this is to regenerate our starting compound because that has to be present for the next round. Okay, so this cycle will turn 2 times for 1 molecule of glucose. And that's just, these are highlighted. Here's our 2 CO2 produced, our 4 different oxidation reactions, there's the ATP. So the big picture for the TCA cycle is this. Basically, carbons, we're bringing in 4 carbon combining with 2 carbons, and then producing these electron carriers and a little bit of ATP, okay? So, you should be able now to finish this row, the TCA cycle row, and again, think about it from the perspective of the cycle turning 2 times. And then after you're done filling in that row, let's take a 10-minute break. I'm not gonna talk the whole time of this class because that's too much lecture. If you do wanna work on your posters a little bit, we can, but we'll kind of finish up a little bit more of this lecture, and then if you're done early, you can either study or work on posters or get your poster printed or whatever it is that you're needing to do. So, yeah. Yes, so positive regulation is when there's low energy levels, like with AM, high AM, then it becomes phosphorylation, so the PDH, the pyruvate dehydrogenase. Yes, rephosphorylation.
PDH.
Yeah.
Yes.